enzyme activity, and today we are talking about where we are able to talk about the lock and key hypothesis, give the advantages, and I gave you an assignment to go and find out the limitations of the lock and key hypothesis. So I hope you guys did, and yeah. So if you did some research, I'm sure you found out that this lock and key assumes the enzyme to be a lock and a substrate to be a key that must have a complementary structure, that must have a structure that complements the active site of an enzyme. So if you assume an enzyme to be a lock, then it means that it assumes an enzyme to be some rigid stuff, too rigid, that it must maintain the shape of the active site. And actually, when you study molecular biology, if you go ahead and study these structures of enzymes, enzymes don't have such a rigid structure. They don't have such a rigid structure. The moment this was, it's okay. So they don't have such a rigid structure. They are flexible, they can move a bit, they are flexible, in other words. So because of that, then there was a need to come up with another theory or another hypothesis which tries to explain how these enzymes work. And it led to the induced fit. Induced fit. Induce means to cause. To cause fit. Fit means, you know fit. You know these days you put on fittings. Fit. So induced fit is to cause fit. So that's how simply I can summarize it. To cause fit. So with this hypothesis, it suggests that yes, it also agrees that the enzyme has an active site and also substrates have a shape. Okay? Yes, it also agrees that enzymes do have an active site. However, unlike the lock and key hypothesis, which assumes the structure of an enzyme to be some rigid shape, this one doesn't assume like that. It have an active site, but the active site actually may not be the exact, may not have that exact shape that substrate must bind to. Okay? It may not have. But rather, when a substrate binds to the active site of an enzyme, then the substrate causes the enzyme, now that is the induce, to undergo some specific configurations or some configurational changes which will lead to some small changes in the shape of the active site, thereby allowing the substrate to fit into the active site of the enzyme. So basically, that's what induced fit. So the enzyme undergoes some configurational changes which will allow the substrate molecule to fit into the active site of the enzyme. And this is really key. Because remember when we were studying the structure of a protein, we saw that yes, the peptide bonds, they are rigid. But the neighboring covalent bonds, the adjacent covalent bonds, they are flexible. So the overall structure of a protein is flexible. So even here, the enzyme is also flexible. Okay? That binding of the substrate to the active site causes the enzyme to undergo some configurational changes. Okay? To undergo some changes which will allow the active site to fully fit into, so the substrate to fit into the active site. And then as the enzyme is undergoing the configurational changes, then you will create strain, you'll be straining the bonds that are formed within the substrate molecule. Okay? And when you strain those bonds, then they will easily break to give the two products, which will account for the enzyme activity. So that's how cool this induced fit hypothesis is concerned. So you can find out the advantages of the induced fit. Okay? Advantages of the induced fit over the rock and key hypothesis, and the advantages of the rock and key hypothesis over the induced fit hypothesis. So if you can compare all these two theories, then you'll be good to go. Let's talk about the enzyme inhibitor. Still, inhibitor, this is an English word. To inhibit means to prevent, to shield. Which other word can we use? So enzyme inhibitors, molecules that prevent, that shield, that reduce. So something of that kind. Something of that kind. Me, I usually like making scientific concept to be kind of lively and easy. So therefore, from that brief view or brief overview, how can you define an inhibitor? So it is that substance which can reduce the rate of an enzyme controlled reaction. You know, enzymes do catalyze. Given reaction A plus B, and the reaction is controlled by enzyme, okay, enzyme Z, okay, to form products maybe AB. So if you want to determine the rate of this reaction, then you measure how many AB molecules are formed in 1 minute. Okay? Yes, if you form 10 AB molecules, divide by 60 seconds, that will be the rate. 10 out of 6. Okay? 10 out of 60, which is 1 point something, 0 point something. Okay? Milligrams per minute. That is the rate. If you are forming 20, divide by 60 minutes, that will be the rate. Now, enzymes do increase the rate of attaining the products, the rate of formation of products, the rate at which products are going to be formed. Now, when I get my enzyme controlled reaction such as this, and then I add certain substance in my reaction vessel, and that substance I've added, it reduces the number of AB molecules that are formed per minute, then that substance is referred to as an inhibitor. So inhibitors reduce the rate of enzyme controlled reaction. Actually, others cut off the reaction completely, that nothing is going to happen, whereas others just reduce. Okay? Okay, it's okay. Sorry for that. Sorry. So now these enzymes, they do interfere with the catalytic process either by completely inactivating or by inactivating the enzyme, hence reducing its efficiency, or completely denaturing the enzyme. Let me say that why. You studied denaturation of proteins. We saw that. So you either inactivate, hence reducing its efficiency, or you completely denature. There, if you denature the enzyme is rendered useless. Let me use that word. It is rendered not only inactive but rather useless. It won't be able to carry out any catalytic process, catalytic reaction. So those are our catalysts. Now, they can be categorized into two types. Depending on how these catalysts, depending on the impact, depending on the effect of a given catalyst on an enzyme. Whereby number one, we can have competitive inhibitors, I think we have it there, and non-competitive inhibitors. I think we also have it there. Competitive and non-competitive inhibitors. Okay? And I'll use this illustration that assuming you and your birthday, you are all students. Okay? You are all students, you all go to the DH, everything is the same. You and your birthday. And you are all eyeing at one babe. You are all same, you all go to the DH. Here you will compete because you are at the same level. Okay? You are at the same level. Everything is the same. So you compete for the babe. And the one with kinda nice words, the one who can frequently meet the babe and pour some good words, maybe he's going to kiss the bride. But if now you the guy, you are eyeing at the babe, and then some other dude maybe from university, now this is a campuser. A campuser, moreover a campuser with money, is also eyeing at the same babe. Then man, obviously you have to be out competed. There is even nothing like competition there. You accept defeat. Okay? And the campuser kisses the bride. There is nothing like competition there. Okay? So those are our two types of inhibitors. So we have the competitive and non-competitive inhibitors. Now, with competitive here, the inhibitor molecule has the exact shape with the substrate molecule. They have exact shape with a substrate molecule. Okay? Now, if the inhibitor has the same shape as the substrate molecule, then it means the inhibitor too can bind to the active site of the enzyme. So therefore, the inhibitor together with the substrate molecule, they will all compete for the active site of the enzyme. They will all compete for the active site of the enzyme because they have the exactly the same shape. Okay? Now, the difference is if the substrate molecule attaches, then the substrate molecule is going to be strained and broken down to form the products. But if the inhibitor binds to the active site of the enzyme, then there is no way how you're going to dissociate the inhibitor molecule to form the products. So it will remain bound onto the active site of the enzyme. And if the inhibitor remains on the active site of the enzyme, then there is no way how this active site is going to bind with a substrate molecule to form products. Hence, the number of product molecules that are formed per minute is going to decrease. Hence, decreasing the rate of a chemical reaction or of an enzyme controlled reaction. But now, however, if I now increase the concentration, okay, of the substrate molecules within the reaction vessel, then the rate of reaction is going to increase. The number of products formed per minute is going to increase. Why? Because you are increasing the substrate molecules, the number of substrate molecules, hence the substrates will outweigh the inhibitors so that you have most collisions between the enzymes and the substrates. In other words, the chance of collision between the enzyme and the substrate molecules is going to be high. Hence, the number of products formed per minute is high. Hence, the rate of reaction increases. Okay? Yes. So, graphically, we can represent it as this. So, you have the rate of enzyme controlled reaction. You have rate here. Okay? And then you have time. What are we studying? Yeah, yes, we have time. Okay? So we can see that the rate, no, we have substrate concentration, not time.
What is the fallacy?
So let's put here substrate concentration. Okay? Concentration. Now, so we can see that then you have the inhibitor. So if the rate of reaction obviously increases, okay, but it increases gradually, or the rate is low because you have an inhibitor. But as the substrate concentration increases, then the rate is going to increase and you attain maximum. So this will be the curve for substrate concentration in absence of an inhibitor, and this will be a curve in presence of an inhibitor. So this is curve, this is in absence of inhibitor, this one is in presence of inhibitor. So if such a curve is given, for instance, you can see that in absence of an inhibitor, it's normal. It will increase and finally becomes constant. And this constancy is because all enzyme molecules are fully saturated with active site with substrate molecules, so no more increase. But in the presence of an inhibitor, then the rate of enzyme activity is retarded. Okay? For the case of competitive inhibitor, the rate is going to be retarded and it's going to be slow. It is going to gradually increase. But when finally the concentration of inhibitors increase, so the concentration of substrate molecules increase, then the rate is going to increase and finally attain that maximum rate. Okay? Why? Because the inhibitor molecules will outweigh, the substrate molecules will outweigh the inhibitors. Okay? Hence the rate of reaction is going to increase. For the case of competitive, inhibitors, because these ones don't affect the enzyme. They do not affect the 3D shape of the enzyme. What they do, they just bind to the active site, and when they bind to the active site, then they prevent the substrate molecules from binding to that active site, hence reducing the rate of a chemical reaction. How about for non-competitive? Here it is, the non-competitive inhibitor is a campus boy, the campus guy. Then the substrate is you. Now for these ones, for years you're busy planning the manifesto, as you're busy planning all kinds of sweet words. For this campus guy, you just come and be like, man, I'm a campuser from Muku, and I have money. And the guy just splashes money. As for you are busy planning manifesto, DH, go to eat DH food, blah blah. For him, you be like, I'm a campuser from Muku and I have money. And the guy splashes money and takes the bride. So for these ones, for the non-competitive inhibitors, what they do, they don't combine to the active side, okay? But they have somewhere on the enzyme. So the enzyme has different sites. So if this is my enzyme and this is my active site, for instance, somewhere on the enzyme, okay, you'll have another site, and that site is called an allosteric site. An allosteric site. Now the competitive inhibitor binds to that site, binds to that allosteric site. It just comes and binds to that site. Now binding of the inhibitor on this site, it alters the active site of the enzyme. That if it was that, it will change the shape and adopts a new shape. And now the new shape does not allow the substrate molecules to bind with the enzyme. Okay? The new shape is completely different from the shape of the active site. Hence, the substrate molecules cannot, sorry for that, the substrate molecules cannot bind to this new shape. And so if the substrate molecules cannot bind, then you don't expect any products to be formed. So you can have a look, okay? You can have a look. So it binds somewhere on the enzyme. It doesn't care about the active site, whether the shape is the same, no, it doesn't even care. Okay? It will bind on the allosteric site somewhere on the enzyme, and the binding of that non-competitive inhibitor on the allosteric site, it alters the shape of the enzyme. So if this campus guy comes, it will be like, man, I'm a campuser from Mu. And then the guy splashes money. So the moment the guy splashes money, then the baby will change. You go there to pour your manifesto and the baby is on her own. Okay? The baby is on her own. She will no longer even give you any attention. You'll be like, what happened? Nothing. How are you? I'm fine. What? I'm fine. So everything will just be berserk. Okay? Everything will just be berserk. And so because of that, then non-competitive inhibitors are really dangerous as far as enzyme activity is concerned. So they can be categorized in two categories. Number one, non-competitive inhibitor reversible, and non-competitive inhibitor or inhibition irreversible. Now with these reversible inhibitors, once an inhibitor binds to the allosteric site of the enzyme and you alter the active site of the enzyme, the extent of alteration, the extent of denaturation is not that big, not that much. So once the inhibitor detaches from the enzyme, then the enzyme can regain its shape of the active site. Okay? And you catalyze the reactions again. And the enzyme works again. However, now for the case of irreversible inhibitors, these ones, once the inhibitor binds, then even if they detach, then the extent of denaturation is going to be so high that the enzyme cannot regain its shape. And because of that, then the enzyme is rendered useless. Unless you produce other enzymes by protein synthesis, then you cannot survive, and of which it's not possible. Not possible. So those are the poisons that you take. So when you take a poison, however, these poisons of usually non-competitive inhibitors, they are heavy metals. Okay? Even. So the poisons that you take, if maybe you are giving a rat and you accidentally take it, maybe you are tired of living and you take some poison. So all those are inhibitors. Whether you are a heavy metal, whether you are an organophosphate, organochlorine, all those are inhibitors. In that when you take a poison, for instance, that contains mercury or cyanide, for instance, now this cyanide is a metabolic poison whereby it inhibits the enzymes in metabolism, in respiration. Okay? Now when you inhibit the respiratory enzymes, then respiration won't occur. And if respiration doesn't, then you cannot produce energy. And if you can't produce energy, then you are useless. You die. Okay? So cyanide is a metabolic poison. All these heavy metals, for example, mercury, eh? Silver, but mostly mercury. This is a poison. This is a non-competitive inhibitor. Okay? For acetyl, not acetylcholine, but coenzyme Q. With respiration, once you produce that pyruvic acid in glycolysis, the pyruvic acid must be incorporated in the Krebs cycle, in the citric acid cycle. But for you to be able to incorporate the pyruvic acid in the citric acid cycle, then the pyruvate must undergo oxidative decarboxylation to produce an acetyl group, CH2, CH3, CH2, that must combine now with the coenzyme Q so that it is fed into the Krebs cycle by combining with OAA to form the citric acid. But for that one to occur, then this coenzyme Q, this coenzyme, it is CoA, not coenzyme Q. This one is found in, sorry for that. This one is found in ETS. This coenzyme A, CoA, it must be there, it must be active so that it can combine with acetyl to form acetyl CoA. But if you are poisoned with mercury, then this enzyme is going to be inhibited. Instead of coenzyme A combining with acetyl group, it will combine with the mercury, preventing it from combining with the acetyl group. Hence, respiration won't take place. So depending on the extent of denaturation by a given inhibitor, then it will determine whether it will be reversible or irreversible. Then another type of non-competitive inhibition is the end product inhibition, whereby in this, the end product formed
Yes, sir.
can inhibit the enzymes, one of the enzymes involved in that process. For instance, if you have a given process, okay, that occurs with different steps, whereby A is converted to B and B is converted to C, and then C is converted to D and D is converted to E, and finally you form your end product as F, for instance. So if F is the end product and F is formed from a number of different intermediates, and all these intermediates are combined, are catalyzed by enzymes. You have enzyme E1, enzyme E2, E3, E4. All these are enzymes, different enzymes, and E5. So the formation of F undergoes, it undergoes through different intermediates. Sorry for that. Then if F accumulates, then if you produce excess of F, it will be a waste. You'll be wasting resources which are not even there. So you first find means of regulating such a process. And the only way of regulating this process is by end product inhibition. And what is end product inhibition? In this case, for instance, the product that is formed, which is F, it can inhibit the enzyme that controls the first step. So that if F accumulates, then this enzyme is inhibited. And if you inhibit this enzyme, then no B is going to be formed. And if no B is formed, then no C is not formed. If no B is formed, yes, then no C is formed. And if no C is formed, then no D. If no D, then no E. And if there is no E, then no F is going to be formed. And if no F is formed, then the F is going to be used by the body and it decreases in concentration. And if the concentration decreases of F beyond a given level, then the F is going to detach from this enzyme, okay, and then the reaction continues. So you can read about, for the end product inhibition, you read about the enzymes involved in glycolysis, okay. Yes, the hexokinase enzyme. You can, you like Google so much, so go to Google, type end product inhibition, you put a plus hexokinase enzyme, you put a plus glycolysis. You'll be happy, you will enjoy this science. Not so? Yes. Yeah, so that's a brief overview of what inhibition is. Let's see some examples of inhibitors. Now in Krebs cycle, we have this enzyme called succinyl dehydrogenase. It is the one that oxidizes succinate isocyanose. Is it isocyano? Okay, succinate. Let me not complicate. To malate. Yes. Then from malate, then you go to OAA. Yes, it is from succinate to fumarate, not malate. Yeah, It is succinate, then fumarate, then from fumarate to marate, and then from marate to oxaloacetic acid. That's how. So the marate formed, the marate and the succinate, all these, they are organic compounds. They are all intermediates as far as Krebs cycle is concerned. So the marate formed, it has the same shape as the succinate. They all have the same shape. So therefore, having the same shape means all these two can compete for the same active site of this enzyme, succinyl dehydrogenase. Okay? So they will compete, whereby the true substrate is succinate, but the inhibitor in this case, it's marate, because if marate attaches, then it won't be converted to fumarate. So if it's not converted, then you won't see the products. Hence the rate is reduced. So that is one clear example of competitive inhibition. Another example is here. We have an antibiotic called sulfonamide or sulfonamide. Now it is a competitive inhibitor for another substance called PAB, para-aminobenzoate. Now this PAB, it is a natural substance that is synthesized by bacteria. Okay? Or it is a natural substance that is needed by bacteria to produce folic acid. Okay? And this folic acid is an important vitamin as far as bacterial survival is concerned. So, no PAB, no folic acid, bacteria die. Enough PAB, enough folic acid, bacteria thrive. Now, scientists have studied the structure of this PAB, para-aminobenzoate, and they have developed another amide, sulfonamide, that has the same shape with PAB. And so they have used this sulfonamide as an antibiotic to treat bacterial diseases. Okay? That you're going to spray these bacteria with sulfonamides, and when you spray them, instead of fixing the PAP, instead of the enzymes that can fix this PAP, they will combine with this antibiotic, and so you won't be able to form the phosphoric acid, and hence the bacteria will die. Then another example of competitive inhibition is between ribop and carbon dioxide fixation. Now this enzyme, it is ribulose bisphosphate carboxylase oxygenase. Let's even add it. Oxygenase. Now this enzyme, it is the enzyme responsible for fixing carbon dioxide. For plants to carry out photosynthesis, they must be able to fix carbon dioxide, and when they fix the carbon dioxide, then you form the 6-carbon compound, which is unstable, that will completely split to form the 3 carbon compounds, the 3GP phosphoglycerate, which is now reduced to form PGAL, phosphoglyceraldehyde, 3-phosphoglyceraldehyde, which can now be converted into glucose and other stuffs. So now for this enzyme ribulose bisphosphate carboxylase oxygenase, the enzyme has both affinity for oxygen and carbon dioxide. Don't mind about this oxygen and carbon dioxide. That in high concentration, of carbon dioxide, then the enzyme fixes carbon dioxide. But also in high concentration of oxygen, then this enzyme can fix oxygen. And instead of photosynthesis, then it undergoes what we call photorespiration. So, boom, you reduce the efficiency. Where you are supposed to form the 3 glucose molecules per minute, for instance, just quoting, you form 1. And if you form 1, then you reduce the efficiency of that enzyme. So basically, that's what competitive inhibition is about. Then with non-competitive inhibition here, the enzyme, sorry, the inhibitor attaches somewhere else other than the enzyme active site. So inhibition in which the inhibitor attaches themselves to the enzyme molecule at the point other than the active site. And this is the allosteric site. So inhibition by causing enzymes to change the shape of the active site, thereby preventing the active site from fitting its substrate. Examples, we have poisons like cyanide, phosphate, insecticides, all those, arsenic, which is a neural gas, heavy metals like zinc, mercury, and silver, all these ones, they are non-competitive inhibitors. Okay? Whereby they do inhibit the enzyme. But they do not attach on the active site, but elsewhere on the enzyme other than the active site. Not so? Yeah, so basically that's what non-competitive inhibition is about. Then with allosteric inhibition, here it is an example of non-competitive inhibition where the inhibitor attaches on the allosteric side. Okay? So enzymes that occur in two forms, i.e. the active and the inactive form. The inactive form is shaped in such a way that the substrate will not fit in an active site. So here, with allosteric enzymes, actually it wasn't inhibition but enzymes. Okay, identity, it's okay. So with these enzymes, they are enzymes that do exist in two forms, the active form and the inactive form. For example, pepsin, which is secreted, or actually most of proteases, they are released in inactive form. Okay, whereby the inactive form, it has to be activated into an active form which can do carry out the catalysis. So therefore, for this to occur, then the enzyme must be able to be activated. And usual activation is by allosteric activators, whereby the activators attaches on the allosteric site and binding of the activator to the allosteric site leads to the enzyme to undergo configurational changes, okay, which will lead to formation of the shape of the active site that is active. Okay, and so the same as allosteric inhibitors, they all do the same. They also bind to the allosteric site, which either lead to forming the shape of the active site that can now carry out the catalysis, or you form a shape that cannot carry out any catalysis. Okay? So for example, you can have a look at this. So you have an enzyme, and that is the shape of the enzyme. That's the enzyme. So the enzyme you must have an activator that will bind somewhere, okay, so that you stabilize the enzyme in an active form, or in a form that can carry out catalysis. Similarly, if an inhibitor binds, okay, if this is the shape, okay, of the enzyme, or if this is the active shape of the active site, and the inhibitor binds, then instead of activating the enzyme, then the enzyme is going to be inactivated, okay, and you form a shape that cannot carry out any catalysis, okay, and you stabilize that inactive form. Then we have, for instance, enzymes that have more than one active site. We have proteins with more than one active site. Now these proteins, they have what we call cooperativity. The active sites are cooperative, whereby with enzymes that have more than one active site, if the substrate molecule binds to the active site, yes, for the first binding it might be hard, it might require some energy, in quotes, it might be hard. But if the first active site binds to the substrate molecule, then the binding of the first substrate molecule induces the overall enzyme to undergo what we call configurational changes, which will expose other active sites to bind with the enzyme, to bind with the substrate with ease. With ease. For instance, take an example of hemoglobin. Yes, hemoglobin is not an enzyme, but it brings out this concept of cooperativity with ease. So hemoglobin, it has 4 subunits with 2 alpha and 2 beta, with a heme group in the middle. And it is that heme group that can bind. Now, these heme groups are inside, they are shielded inside, they are not easily exposed. So it is hard for oxygen to access these heme groups. It's really hard. But once the first oxygen, or once the first group binds to oxygen, then it will induce, or it will stimulate other groups to be exposed. And binding of the second exposes the third, and the binding of the third. So in other words, it creates a cascade effect, where binding of this opens this, and binding of this opens more of the other. So it is hard for the first to combine, but once the first combines, then it becomes easy for the third, the second, third, and fourth groups to combine. Hence, when you try to plot the percentage saturation of hemoglobin with maybe oxygen, you find that you get such a curve. You get a sigmoid curve, okay? Whereby it is hard, but once the first heme group combines, then it becomes easy for other groups to combine. So that phenomenon is what we call cooperativity. So binding of one substrate molecule to the active site of one subunit locks all subunit to the active conformational change. So it opens other active sites, and the other active sites are willing to receive a substrate. So that phenomenon is what we refer to as cooperativity. They are cooperative. Then you can read about feedback inhibition. Yes, this is an assignment that I gave. I told you to read about hexokinase and glycolysis and end product inhibition. Just go to Google, type end product inhibition plus hexokinase plus glycolysis. They will give you data, okay? And you read about this. Or you can use the textbooks that I posted in our classroom, okay? Yes, you can also find it. Or you can watch the video by the where I talked about glycolysis in respiration. I talked about it. Okay? Yes, so otherwise I think I'm going to stop here. Okay? Yes. Yeah. If there are no questions then it's okay, we can call it a day.